AP Biology, second quarter review, part 7. Remember that in meiosis we have 2 rounds of cell division, and if you see a 1 or a 2 after a phase of like a prophase or a metaphase, that's referring to meiosis only. Remember, during mitosis, there's only 1 round of cell division, so there's no real point in having a 1 or 2 after it. It's just prophase, metaphase, anaphase, telophase. So that's one thing to remember. During meiosis 1, you do need to know that the first cell division separates to homologous pairs of chromosomes, going from diploid to haploid. That's the main event in meiosis 1. During meiosis 2, we're separating out just the sister chromatids that are attached at the centromere. We're going from 1 chromosome consisting of 2 sister chromatids to 1 chromosome consisting of 1 strand of the chromosome, just like mitosis. In fact, really the only difference between meiosis 2 and mitosis is that mitosis you're going from diploid to diploid, in meiosis you're going from haploid to haploid. So here we have a comparison of mitosis and meiosis. Make sure that you know this. Mitosis is 1 cell division, meiosis 2. Daughter cells in mitosis are genetically identical to parent cell, they're clones. In meiosis, they're different. In mitosis, you produce 2 cells, in meiosis 4. Mitosis, diploid to diploid, meiosis, diploid to haploid. Mitosis produces somatic cells, body cells, and cells for growth and repair. Meiosis produces sex cells, gametes. Mitosis has no little crossing over event, or swapping little pieces of chromosomes during prophase 1. In meiosis, we do have that piece of chromosome being swapped over during prophase 1. So we have 3 sources of genetic variability, and this is important to know. The first one is independent assortment. These chromosome pairs assort independent of each other. This blue first chromosome could assort with either this blue second chromosome or the red second chromosome. They assort independent of each other. And that's Mendels' second law of genetics, the law of independent assortment. That's going to create a lot of diversity in the offspring. That's 2 to the 23rd power, or 8 million possibilities of mixing and matching 23 pairs of chromosomes in humans. During prophase 1, and you do need to know this, we have the crossing over or little swapping of pieces of DNA between the homologous chromosomes, the non-sister chromatids of homologous chromosomes. And this happens during prophase 1. What we have is a tetrad, 1, 2, 3, 4 chromosomes. The verb that describes this event is called synapsis, the crossing over event, and this happens, this little area that crosses is called a chiasma. So those are some words associated with prophase 1 and crossing over. And we also have random fertilization. We have a random egg fertilized by a random sperm, so we have 2 sources of genetic information here, and that's also going to increase the variability in the offspring. Lots of differences in the offspring because of all this mixing and matching going on. So how does this affect natural selection? Why do we have sex instead of asexual means? Well, if all the members of a population are the same, produced by cloning asexually, there's less chance that a positive trait in the population will be selected for. If you're a white moth on a white background and you produce clones, they're going to be white moths. And if the environment changes to a gray background, white doesn't, blend in as well. You're more likely to get eaten, and your brothers and sisters and everyone else is going to get eaten as well, because there's no diversity. There's no variability as a result of sexual reproduction. In moth populations, though, we do have a sexually reproducing creature that increases the variability, the spreading of different traits throughout the population. Now, even if the background's white, there's still going to be some black and gray moths being produced. Now, those moths are not going to have an advantage on a white background. However, if the environment changes, let's say we have a gray background, well, now there's a chance that some of the offspring have a survival trait that can be selected for. These guys are more likely to survive because they hide a little bit better. The other ones get eaten. These guys pass on their traits, and eventually you have a population of moths that blends into their environment. And that's basically what natural selection is all about. So that's why we have sex on the planet, is to increase the variability of the offspring, to give the offspring a better chance to survive if the environment changes. And as we know from the history of this planet, the planet has changed quite a bit over time. Now, there are differences across kingdoms. When we talk about plants, we're going to talk about how they have a very large haploid part of their life cycle. But most animals have a fairly large diploid part of their life cycle and a very small haploid part of their life cycle. But that's not true for every organism. There are some differences between kingdoms as far as haploid and diploid. For example, this little heart-shaped part of the fern's life cycle is all haploid cells. All the cells in there are only one set of chromosomes. We'll talk more about that fourth quarter. Mendelian genetics. Mendel is considered the father of genetics, and he was the first to quantify and use the experimental method to determine the laws of heredity. And he figured out that if you cross a plant and there's different dominant recessive traits that are seen in the offspring, and they skip generations. The P represents the parent generation, F1 represents the children of the parents, and then F2 represents the children of the children. So make sure you know what those words mean when you see problems on that. So we have two copies of a gene, and the different versions of a gene are called alleles. So we have two of every chromosome, and on those chromosomes are sequences of DNA that codes for a protein called a gene. And if you have one version of the gene, if you're a pea plant, you might produce purple flowers. And if you get a slightly different version of the gene, probably a few mutations in there, you might produce white flowers. So you could have two purple alleles, two purple versions of the gene for flower color, two white alleles, or a purple and a white allele. And this is going to be useful when we talk about how Punnett squares work. Punnett squares are used to predict offspring, and here we have the two different alleles on the homologous chromosomes, big P, little p. This is a diploid cell in a plant, got two sets of chromosomes. And when this plant, let's say it's male, the male part of the flower, is producing gametes, sperm, and that's going to be haploid, or one set of chromosomes. So we're going from two sets to one set. Now in the process of meiosis, we can either produce sperm with a big P, in this case, or a little p. And these are the two possible gametes alleles packaged up inside that can fertilize an egg. The female here is the same genotype, big P, little p, and the eggs can either have a big P allele or a little p allele. And when we do the cross, this is the possible offspring. It's not automatically going to have 50% big P, or big P for 25%, big P, little p for 50%, and little p for 25%. This is a probability, just like a coin toss. If you flip a coin 5 times in a row heads, doesn't mean it's going to be tails next time. The coin has no memory. Every offspring being produced in this cross has the same percent chance of getting each one of these genotypes. And remember, a genotype is a description of the genes of the organism, and it's represented by letters. The letters just represent the DNA sequences on the chromosomes that code for a trait. Here we have a monohybrid cross. Mono means one, so we're just analyzing one trait. And the genotype ratio for a heterozygote, a monohybrid cross, two heterozygotes, one of each allele, is 25%, 50%, and 25%, or 1 to 2 to 1. Now there's two ways to get the purple phenotype, big P, or big P, little p. So there's a 75% chance of getting the physical phenotype characteristic of purple flowers. That's what phenotype means, the physical characteristics. And 25% chance of producing white flowers. Keep in mind that there's only one way to make the recessive phenotype, little p, and there's two ways to make the dominant phenotype, big P, and big P, little p. So in a heterozygote cross, we always have a 3-to-1 ratio for one trait. Here we have a test cross. Oh, we don't know what that purple flower was. You just look at a purple flower, you're not going to be able to tell if it's big P, or big P, little p. So if we cross it with a white flower that has a known genotype, little p, only one way to make white flowers, if the parent was big P, and crossed with little p, then all the offspring are going to be big P, little p, and they're all going to look purple. So this is one way we can try to figure out what the genotype of the dominant phenotype parent is. Over here, if that purple flower is big P, little p, and we don't know until we do the cross, and we cross it with a white flower, then we're going to get a 50% purple phenotype and 50% white phenotype. Now keep in mind that if you get just purple flowers, that really doesn't help you out. However, if you get any white flowers from this cross, then you know the parent was big P, little p, because the only way to make white flowers is if that purple parent was big P, little p. And that's what a test cross is about, trying to figure out the genotype of the dominant phenotype parent. All right, Mendels Law of heredity. Remember that log of segregation. The homologous chromosomes separate out during meiosis 1. One chromosome goes one way, the other one goes the other way. The law of independent assortment is referring to two or more traits, not one trait like segregation. And these two or more traits are found in different homologous pairs of chromosomes, and they sort independent of each other. Chromosome pair 1 separates independent of chromosome number 2, pair number 2, and any of the alleles for different traits on those other chromosome pairs are also separate and independently assorted during meiosis. Both happen during meiosis 1. Remember that for monohybrid cross, we're talking about one trait, for law of segregation, just traits on the homologous chromosomes. For law of independent assortment, we're talking about two or more traits. And here we have the one of each allele for a trait being separated during meiosis 1, law of segregation. This chromosome goes one way, this chromosome goes the other way. Law of independent assortment means that if other traits are on this other pair of chromosomes, these chromosomes assort independent of these. The red can be packaged up with the red or blue. That's independently assorted, and that's the second law. So the alleles for different traits don't usually travel together unless they're on the same chromosome. So what ends up happening is we find out that the types of gametes that we make are independently assorted, and we have to figure that out when we do our 16-square Punnett squares. This is the correct answer. The data from our crosses in a dihybrid cross end up supporting this method of independent assortment of alleles for different traits. Here we have a dihybrid cross. Remember to figure out the alleles, and this is kind of, or figure out the gamete possibilities. You package up the first with the first. Big Y, big R is one possibility for the gametes. Big Y, little r is the second possibility. Little y, big R is a third possibility. And little y, little r is the fourth possibility for gametes for that first parent. And this parent is genetically identical, so that's the same possibilities. On a 16-square Punnett square, you just add them together, big Y, big R, and figure out what kind of phenotypes you're getting. In a dihybrid cross of heterozygotes, you always get a 9:3:3:1 ratio that you're going to need to memorize of phenotypes. You don't have to know the ratio of genotypes. All right, here's another diagram of that. All right, incomplete dominance is like when you mix paint together. Think of it as like mixing red paint with white paint to get pink paint. Now, things like snapdragons, pictured here, and carnations have something called incomplete dominance. So you have the red allele, and if you get two red alleles, you're red. If you get two white alleles, you're white, just like regular dominance recessive. However, if you get a red and a white allele, it doesn't have red dominant to white is not dominant to red, you produce a blending of the two. Now, that's going to be different from codominance. A blending of the two, like paint mixed in a mixer, is called incomplete dominance. And this is our example. This C just refers to the color gene on the chromosomes. We can leave out the C there. We just use R's and W's if we wanted to. Notice that we didn't make it a little w because it's not really recessive to the red. And if you look at our offspring, the genotype, we'll separate out the genotypes for homozygous dominance. Two of the dominant allele, in this case it's not really dominant. The heterozygotes, one of each allele, appear pink instead of red or white. Codominance is when you express both alleles separate and distinguishable from each other. Now, the ABO blood groups are one example, but an easier one to visualize are chickens. When chickens have the black feather allele and the white feather allele, they don't make gray. That would be incomplete dominance. You produce a chicken with black and white feathers, distinct from each other. That's because those alleles are codominant, and they're both expressed. Now, the ABO blood groups are also codominant. If you get the A and the B allele, you're going to have both A and B glycoproteins on the outside of your red blood cells separate from each other, and that's why it's codominant. This is also an example of multiple alleles. There's not just two alleles for blood types, but there's three alleles, A, B, and basically a blank allele, or an allele that doesn't give you any glycoprotein. Both A and B alleles are dominant to the blank allele. And the only way to have type O blood is to have two of these alleles with nothing on it, blank, and that produces nothing on the outside of the red blood cells. Oligosaccharides are basically kind of similar to glycoproteins. They're used for cell recognition, and we'll talk more about that fourth quarter. Here we have the blood types. There's two ways to get type A blood, either AA or A blank. The I is just represented its blood type. You can ignore that. B can either be BB or B blank. AB, only one genotype for that, AB. And O is blank blank. As far as how these blood types react with other universal donors, things like that, we'll talk more about fourth quarter. But you should know these alleles and be able to do Punnett scores with these alleles to figure out offspring. All right, at this time we're going to stop part 7 of our AP Biology review for second quarter.